Aerobic Respiration
by Ethan,
Daniel,
Matthew,
Sabina,
and Sophia. This is the link reaction.
This is the cristae, this is the outer membrane, this is the inner membrane, and this is the matrix where the link reaction happens.
Let's start right after glycolysis.
We've got pyruvate, a 3-carbon molecule, ready to enter the mitochondrion. 1 carbon is released as CO2. NAD plus grabs electrons to become NADH. And what's left is a 2-carbon molecule, acetyl-CoA.
Dun dun dun. Here we see the Krebs cycle. The Krebs cycle is what is used by the chloroplast to allow the cell to have energy. Not produce energy, because energy is neither produced nor destroyed.
Wow.
Thank you for that wonderful energy, Sophia. Which is neither produced nor destroyed. The first transfer that we will see in this is from 6 carbon to 5 carbon, which goes along with side by side. What's happening is NAD is moving to reduced NAD, and this produces CO2 as a byproduct for plants. After this occurs again, but instead of 6 to 5, it is 5 to 4 carbon. Again, NAD is moving to reduce NAD, and CO2 is being produced. After this, it is 4 carbon to 4 carbon. Instead of NAD to reduce NAD, we are moving from ATP to ADP as our simultaneous reaction. This does not produce carbon dioxide because it's sucky.
How is he expecting this to describe it? You don't even know.
After this, again, we're moving from 4 carbons to 4 carbons, and besides this, we are moving from FAD to reduce FAD. Again, no carbon is being produced. After this, we are adding H2O to the mix. Again, moving from 4 carbon to 4 carbon. We are then again adding NAD, which is shifting to reduce NAD. And finally, the culminating moment of this cycle, which there isn't really one because it just goes on and on. Culminating moment is finally where we are moving from acetyl to the acetyl co-transporter, and we're moving from 4 carbon to 6 carbon, from oxaloacetate to citrate. Thank you.
So the reduced NAD releases energy, which pushes the protons up through the proteins, and it continues to go through all the proteins, pushing protons up out of the proteins.
And at the end, oxygen is used as a main electron acceptor, and then which allows it to accept the other protons, and it creates water as a byproduct.
Energy is needed to pump protons across the intermitochondrial membrane as they are moved against the concentration gradient. Though the energy is not lost, but it is stored as the proton gradient and then used to make ATP. The ETC, which is right here, generates and maintains the proton gradient across the intermitochondrial membrane by pumping protons across the membrane using energy from the electron flow. The protons move from lower concentration gradient to higher concentration gradient in the intermembrane space to decreased concentrations in the matrix of the mitochondrion.
Next we will be explaining chemiosmosis. The drum-shaped part of ATP synthase in the membrane has identical subunits, each with a binding site. It also has, one allows for protons to enter and bind to the subunit. Others allow previously binded protons to leave. The drum has to rotate as two half channels are not aligned. Each proton goes around for almost a full rotation. Tightly connected to a stalk when projects into a matrix. And due to the tightness, the stalk rotates at the same rate. This is known as the rotor of ATP synthase. The stalk is surrounded by a globular part of ATP synthase, which has a ring of alternating alpha and beta subunits. Beta subunits have active sites for phosphorylating ADP to ATP. When the stalk rotates, beta goes through conformational changes. ADP has a phosphate group binded to the active site. Then phosphate links ADP to ATP. ATP is released into the matrix, and rotation is prevented by the rotor arc.